Today I'm going to be presenting some new features in LaserGene for aligning multiple genomes in LaserGene, and then using that alignment to transfer annotations between genomes. To kick off today's webinar, I'll just be giving you some brief background on DNAstar as a company, as well as our LaserGene software. And then, as Sharon said, the majority of the webinar will actually consist of a live software demonstration where I'll show you tools for multiple genome alignments and annotation management. So a little bit about DNAstar as a company. We were founded in the lab of Dr. Fred Blattner at the University of Wisconsin, and we've been in business for over 30 years. Our original commercial efforts focused on DNA sequence analysis and molecular biology, and specifically tools supporting Dr. Blattner's work sequencing the E. coli genome. And as the fields of molecular biology, genomics, and bioinformatics evolved, so have we. So we have expanded on our molecular biology offering, and we've also, over the past several years, added several tools for next-generation sequence assembly and analysis. And then more recently, we've added tools for protein analysis, including protein modeling and structure prediction. So this long history and widespread use of our software has made DNAstar the leader in citations in peer-reviewed journals. In fact, over the last 31 years of our history, we've actually been cited in more peer-reviewed journals than any of our commercial competitors. And if you're interested in learning more about publications citing DNAstar software, we have a nice section on our blog on the DNAstar website. About once a month, we feature some recent publications citing our software. And more recently, we've been featuring publications by topic. So you'll see more of these posts coming soon. But for example, if you wanted to see how other researchers are using our software for viral genome assembly, for example, we've got a whole post here with some recent publication examples. Another great resource on our blog is our customer interviews section. Here you can see some short Q&A with researchers using our software, just to give you an idea of the kinds of projects you can do with DNAstar software. DNAstar is a global software company. We're headquartered in Madison, Wisconsin, and we have a full support team in-house to assist you with any needs you have regarding the software. And our support team also works closely with our development team, which means we can use any feedback you give us to make our software even better. And to supplement this in-house support team, we also have global distributors around the world. So a little bit about our software. We recently released LaserGene version 13. And LaserGene supports a wide variety of workflows, including molecular biology tools, as I mentioned. So our molecular biology suite supports a variety of workflows for virtual cloning and primer design, multiple sequence alignment, which we'll be looking at today, phylogenetic trees, as well as Sanger sequence assembly and analysis. Our LaserGene Genomics suite integrates with the molecular biology tools and also allows you to perform next-gen sequence assembly and analysis, including gene expression analysis and variant detection and analysis. And then our structural biology suite provides tools for protein sequence analysis, secondary structure analysis, and 3D protein modeling. All of this software is available on the desktop computer as well as on the DNAstar cloud, providing you flexibility in accessing and sharing your data, as well as access to powerful hardware on the cloud, for example, for large next-gen sequencing assemblies. We also have flexible licensing options, so whether you need a single license, a shared license for your group, or an institute site-wide license. We can work with you to find the solution that best meets your needs. So that's just some very brief background on our company. At this point, I'll get into the software and show you some of the new features LaserGene has to offer. So you'll see here on my screen, I have the DNA Star Navigator open. And the DNA Star Navigator is your resource for both getting started with your sequence analysis projects and also learning more about the software. So you'll see we show all of our applications here. They're grouped by workflow, so you can see molecular biology here at the top, and this is where we'll be focusing our attention today. You can also access our genomics and transcriptomics tools and our structural biology tools. You can also access the DNA Star Cloud resources from here as well. And as you hover over each application, you'll see a brief description comes up, as well as links to videos, tutorials, and demo data. So as I said, this is a great way to just learn about DNA Star software, but also for experienced users to jump in and start their individual projects. So to start off today, we're going to be looking at some multiple sequence alignments and specifically multiple genome alignments. So for that, we'll want to launch Megalign Pro. And Megalign Pro is designed to make it very easy for you to align multiple DNA, RNA, or amino acid sequences, usually with just the click of a single Run Alignment button. And the main focus of today's webinar is looking at whole genome alignments. And Megalign Pro is powerful enough to actually do those on a desktop computer. Before I get into that, I just sort of want to show you some basic Megalign Pro functionality. So we'll do some smaller gene alignments before we do the full genome alignment. So with any project in Megalign Pro, you can use the Add Sequences button to add your data. In this case, I'm going to be adding some Megalign Pro demo data. And you can use this Add Sequences dialog. You can also do drag and drop to add your data. And so once I've added my sequences, you'll notice at the top we have this unaligned sequences view. And here my sequences are shown as gray blocks, and the gray color indicates that they're unaligned. And you'll notice as I'm scrolling through this top view, I can see the actual sequence data in the sequence view at the bottom. And from either of these views, you can expand the sequences to expose any annotations that are on your sequences. There's also some tracks, including rulers and information about the quality of the alignment that you can add to these views as well. So to see that, we need to actually perform an alignment. And so all you need to do to perform an alignment in Megalign Pro is either use this Align toolbar button, and you'll notice as I'm hovering over it says Align using muscle. I can change that to use any one of 4 alignment methods. So we have Clustable Omega, MAFT, Muscle, and MAUV. MAUV is used for whole genome alignments, and since I've added amino acid sequences here, that's disabled. So for this particular data set, we can choose from 3 methods. If you want to adjust any of the default parameters before running the alignment, you can choose Align with Options. So that's going to bring up a dialog here where you can adjust the alignment parameters. And the available parameters will depend on the selected alignment method. So I'm just going to align using muscle. And you'll see that completes rather quickly. And now at the top of the window, my sequences are shown as aligned sequences using green and gray bars. And so the green segments are portions of the sequences that align to one another. And the gray areas, for example, if we hover over this larger gray area, those represent gaps in the alignment. And you can see those down here in the sequence view as well. Now, at any point, if you want to add sequences to your alignment, you can do that. So for example, here, I'll just show you I can drag and drop 2 more sequences onto the file. And you'll notice Now I have some extra panes here. I have my alignment overview, and now my unaligned sequences are shown here, again, as those gray unaligned blocks. And that's also repeated down here in the sequences view. We have aligned sequences as well as unaligned sequences. And so to merge these new sequences into the alignment, all I need to do is click this Merge Unaligned Sequences button. And by default, that's going to rerun the alignment using the same method and parameters used before. So you can click Align, and you can see very quickly my new sequences are added to my alignment. Another useful feature is the ability to rerun an alignment, either using different parameters or using a different method. So for example, we can realign these sequences using the MAST algorithm and then display the results in the same manner. Additionally, to analyze your alignment results, you can view a distance matrix. This shows you pairwise distance values between each set of sequences. You can also view a phylogenetic tree. And you can save this tree out directly from Megalign Pro. So for example, if you want to use this image in a publication or presentation, you can export this. You can also export this as a tree file, going to the Export Data command and selecting Tree. And so you can export that as a Nexus or a Nuix tree file format. One additional option that's helpful, especially if you're looking at a phylogenetic tree for presentation, is the ability to rename these sequences as a batch. So to do that, I'll just right-click in this Sequences panel and choose Sequence Label. And you'll see right now we're using the default name, and that can vary depending on the file. If you want something a little bit more consistent or perhaps a little more human readable, you can add in some additional fields. And as you add in the fields, you'll get an example preview name here at the bottom. So you can remove and add things as needed to get to the name that you want. And then click OK, and now you notice all my sequences have been renamed. And so these labels will also show up on your phylogenetic tree here. So you can adjust those names as you need to. You can also adjust them individually if you don't want to use the same convention for all of your sequences. So that's just some basic usage in Megalign Pro. The next thing I want to show you is a full genome alignment. And for this example, let's look at, I have here 2 E. coli genomes. So I'll just drop these onto my new project. And those sequences, as they're loading, I'll just explain a little background. The first sequence I'm loading is a FASTA file with the DH10B substrain of E. coli K12. And the second sequence I've added is the K12 MG1655 strain. And this sequence is a GenBank file, so it's fully annotated, whereas the first sequence is a FASTA file. It doesn't contain any annotations. So you might have this situation, for example, if you've done a de novo assembly and you have your consensus sequence and you want to export that out and align it to another genome sequence to compare annotations or transfer annotations. So if I expand this top sequence, this FASTA file, you'll notice there's no annotations here. A really nice feature in Megalign Pro, especially when you're working with larger sequences like full genomes, is the ability to zoom in and out. So if I zoom way out on my sequence view, I can actually get a better picture of the annotations on the file. Similarly, you can zoom into the overview and expand features there. So at this point, the goal here is going to be to transfer these annotations from MG1655 to the DH10B strain. Before I can do this, though, I need to align these 2 genomes to one another. And again, I'm going to just quickly modify my sequence labels To get something a little bit more human readable, we'll try the description. And so here we can see the E. coli K12 substrain names. Okay, so to perform the alignment, it's just as easy as before. We'll go to the Align toolbar and select Align using MAUVE. And the MAUVE alignment method is unique to whole genome alignments, and it's able to surmount some issues that make these kinds of alignments very difficult for other alignment methods. So for example, MAUVE is able to deal with large insertions and deletions, transpositions, duplications, inversions, things like that that are unique to whole genome alignments that alignment methods geared towards smaller sequences really can't deal with. So that alignment takes just a few minutes. It's almost finished. In the interest of time, I'll show you an alignment I completed earlier today. And so what the MAUVE algorithm does is it breaks the alignment into smaller sub-alignments, and those are represented in the results as locally collinear blocks is what they're called. And what that means is these are blocks of sequence that are relatively free of any major rearrangements or inversions, at least internal to that block. And so if we zoom in a little bit at the overview level here, we can kind of get a picture of where these blocks are on the two genomes. And not surprisingly, we know these strains are very similar, so we only see just a few large blocks and then just a couple smaller little rearrangements and inversions. So the blocks of the same color are the matching portions between the two genomes, and then each set of blocks is connected by a vertical line. So for example, here we have this pink block. You can see there's a rearrangement there between the two genomes. And if we double click that block, it's going to orient the strands so those blocks align, and then you'll see the other blocks then become out of alignment and are now out of focus. So if I scroll over this area, my sequences view shows the sequences grayed out because that's not the actual alignment there. So I can view the alignment only within the active block. If you see situations where one of the blocks is below this horizontal line, that represents an inversion. So if I double click that, the program will actually flip strands to get those two segments in the same orientation. And you'll notice too, if I expand the bottom, we can see the rulers on the two strands here. And again, notice we have annotations on MG1655 and not on DH10B. So at this point now, we've got the alignment completed and we're ready to map the annotations. There's several ways to do this. You can either map all of the annotations, you can filter for certain types of annotations, either based on something like a gene name or how well the sequences match in that region. You can also map just very specific features. If you know there's a gene that you want to transfer between the sequences, you can locate that and map that. So that's what I'll show you first because that's kind of the simplest example. And I'm going to zoom out a little bit here just to get a little bit of a better picture of my annotations. So here, for example, I'm looking at a region of the alignment that contains the LAC operon and MG1655. And we can see we have pretty good alignment here. If we look at this gap fraction graph I've shown, there is a slight, there's an area with a gap here. But overall, these regions appear to map pretty well. If we zoom in, we can see the actual sequence alignment. So let's say I manually just want to move one of these LAC genes from MG1655 to DH10B. All I have to do is right-click on the feature and then select Map Feature to Top Sequence. And that's just going to instantly map that to my DH10B sequence here. In many cases, you may not know exactly where the gene is or you don't want to find it this way. there's some options for filtering based on gene names. So that's what I'll show you next. So what I'm going to do is select my bottom sequence, my annotated MG1655, and then go to Features, Map Features to Top Sequence. And here you'll see several options. You can either map all features, features matching a certain search criteria, or features excluding a specific search criteria. In this case, I'm going to look for features matching, and we'll map the CDS features. And if we want to map, say, all of the CDS features from the lac operon, we can filter and look for a gene name that contains the term lac. And then I'll use an asterisk as a wildcard there. So that should find all of the 4 lac genes from MG1655 and copy them over to DH10B. If we click Next, we're presented with some additional mapping options. By default, the program will look for features that map at 80% coverage and 80% sequence identity. You can adjust these to be more stringent or more relaxed. You can also choose a genetic code for your translation. That's something that would be contained in, say, a GenBank file that you export downstream of the mapping. And then there's some additional options for reporting. For now, I'll keep these up to their defaults, and we'll click Next. And then you just need to set a name for this mapping project. And what that will do is tell Megaline Pro where to save out the report of mapped genes. So I'll click OK, and then it's going to search for and then map these features. And you can see the progress at the bottom. It's mapping the 4th feature and writing a report. And after the mapping is complete, you'll be presented with a link to the report. The report comes out as a tab-separated text file, so you can open that up in Excel. I've got one I ran earlier today open here. So here you can see the results, the 3 genes in the lac operon, and then the regulatory LAC-I gene are all copied over. And you can see the percent coverage and identity for each gene. You can also see if there were any gaps. Again, remember we saw that gap in the LAC-Z gene. That's showing up in our report here as well. And the source and new coordinates of the genes. You can also see these directly in Megaline Pro. If we go back there, close this. And so now you can see here, excuse me, clicked too far. Here in the top sequence, we've got the 3 genes in the lac operon and the regulatory gene, as well as the original gene feature we copied over manually. So that's the second way you can look for genes and CDS features to map over. The third way is to map, say, all of the features from one genome to another, as long as they match at the threshold you set. So that's what we'll look at next. Before we do that, I'm just going to strip these features from the top sequence. As you continue mapping, it will just add annotations. So if we didn't remove those, that would duplicate those features. So now we've stripped the features out. And again, we'll just go to select our source sequence and go to Features, Map Features to Top Sequence. And here we'll choose All Features. And let's say we want to have a more stringent mapping. We can set the feature coverage and identity thresholds to 95%. We can use the bacterial genetic code if, let's say, we want to look at the translated amino acid sequences later. We can choose the correct genetic code. We can also include specific sequence changes in that output report we looked at. And another useful feature is the ability to make a report with all of the unmapped regions. So we can look for intervals of at least 100 base pairs that don't map between the genomes. And that may be something you want to look at to see areas where you may want to do a BLAST search or try mapping to another genome, for example. So I'll click next. Again, you're just going to save out your report. That workflow is the same as what we just looked at. So I'm just going to jump over to my results. So here, I ran this earlier today. You can see here's a list of the unmapped regions. So you get sequence coordinates of every region in the genome of MG1655, or excuse me, DH10B, that wasn't mapping to the MG1655 genome. So you can look at those regions in more detail. And then you can also look at the feature report. And this is really nice. Here you'll see, it's a lot more features because it's looking at the whole genome this time, not just for the lac genes. And for each feature, you can see was the feature mapped or not mapped. So this source feature was not mapped. You can see the percent identity. You can see many genes are identical. You can see if there was a substitution or a synonymous change. So here, for example, if you wanted to filter to look for features that mapped but contained a non-synonymous or nonsense change, you can quickly filter onto that criteria and locate those genes of interest. Finally, if you then want to save out your file, so let's take a look at, here's the genome alignment with the mapping completed. The next step usually is going to be to save out this DH10B sequence with the new annotations. So to do that, you can export the data, choose sequences, and then export as a GenBank file. And so then you can use that GenBank file for, say, a templated genome alignment, or you can open that GenBank file in a sequence editor, for example, our SeqBuilder application, and do any micro-editing, view the annotations, or edit any annotations here as well. We're running a little bit short on time. I just want to briefly mention we do have another application called SeqNinja. And SeqNinja is based on a programming language we wrote in-house here at DNAstar, which is really geared towards molecular biologists and bioinformatics specialists. It's a really powerful program that allows you to manipulate and batch edit large sets of sequences or large genome sequences. So if you have some systematic annotations or edits you need to make, you can use SeqNinja for that. And so SeqNinja also provides a wizard, much like the Megaline Pro options, to set up a feature mapping. And so the wizard looks a little different. You have to browse to your sequence files, but you can set up that mapping, including the MOV alignment right here in SeqNinja. And that'll build a script that you can use as a template for other mapping. So if you have a lot of these projects that you need to do, you may want to look at using SeqNinja to do some of the mapping. Additionally, SeqNinja has other tools. You'll see here's some templates available here. One feature I thought would be useful to show for this example is, let's say you want to take your results, so that GenBank file we saved out with all of the map features, and then you want to extract the CDS features. Perhaps you want to look at the CDS features that didn't match 100%, so you could look for features with an identity in the 90% range. I should mention that when Megaline Pro or SeqNinja do the mapping, they actually annotate the features and specify, let me show you here. You'll see this note at the bottom that specifies that it was mapped from the MG1655 genome, and it shows you the coverage and percent identity. So then over here in SeqNinja, you can actually use that information to filter on only those genes that matched at 95 to 99%. So then extract all those genes, and then maybe you want to translate them into amino acid sequences. You can set up a program to do that all right here in SeqNinja, and it'll do it in one step for you. And then, so in addition to looking at your whole genome sequence, you could look at the resulting translated CDS features. And then you could, look at that amino acid sequence in more detail, perhaps come into our Protein 3D or structural biology suite and look at the secondary structure predictions for that amino acid sequence, or even predict the 3D model to show that protein and see if there might be any important structural differences in the CDS, in the gene, in the new sequence, in the new genome sequence. That's a whole nother workflow that I'd love to show you today, but we are just about out of time. So at this point, I'll take any questions, but we will have other webinars on our structural biology tools soon.
Okay, thank you so much for that presentation, Katie. We did have a lot of questions come in during the demo. I answered a few on the chat. How many questions would you like? Do we have time for?
Yeah, we have time for a few. If people want to stay and listen to the answers, they're certainly welcome to. But we do want to respect your time. So if people need to leave, we can certainly follow up with you later. But I can take a few questions now.
Okay. So the first question is, why would I use annotation mapping rather than performing a templated assembly?
Yeah, that's a good question. And there's various reasons. One reason is that in most cases with a templated assembly, you would have to choose one reference genome. And so maybe you've done a templated assembly, but you have some areas that don't match to your reference genome that you want to map to another genome. Or for example, here I have the DH10B sequence aligned to the Phi80 phage. So you could look at maybe and see if there's any annotations from the phage that you might want to map over that you didn't get from your templated assembly to, say, MG1655. Another reason may be that you're bringing in data, maybe you have a FASTA file from another source that wasn't part of an assembly, or, maybe you got it from a colleague. There's various reasons you might have an unannotated file. So we support files that came from our DNA Star pipeline, as well as files from outside sources.
Okay, thanks, Katie. And the next question is, why would somebody want to use SeqNinja for mapping features rather than Megaline Pro?
Yeah, that's a good question, and I know we kind of had to do the SeqNinja part fast. There is a nice resource in the SeqNinja help. So here I have the Map Features Help dialog open, and it's got a nice little table here that explains sort of the differences in Megaline Pro and SeqNinja. And one of the main differences is that SeqNinja also allows you to map annotations from a VCF file. You need to transfer coordinates between genomes. SeqNinja lets you do that. Additionally, I didn't show this, but SeqNinja will create a table of all the annotations. And so you can choose to edit that table. Let's say you want to add some notes or something. You could edit that table before you actually map the features onto your new GenBank file.
Okay, thanks. That was a good explanation. The next question we have is, how closely related do organisms need to be in order to transfer features between them?
Yeah, that's a good question. So the answer is kind of, it depends. You do have to perform this alignment first. So I guess the most basic answer would be that your genomes have to be, closely related enough to actually align using MAUVE. You saw the default stringency was 80%, so not only do the genomes have to align, but the features would have to match, 80%. You could lessen the stringency there, but at a certain point, those results may or may not be meaningful.
Okay, and our fourth question, can I transfer annotations between sequences that were aligned using a different method, like muscle?
You can, yes. So in this example, we were doing a whole genome alignment, and so that's why we had to use MAUVE. But certainly, in one of my earlier examples where we used muscle and MAFT, you could transfer the annotations from these results as well. The key is just that you have to have completed an alignment first.
Okay, and we have one final question that I think we'll be able to get to. Well, we're a little over time right now, but let's try for one more. And that is, can you add annotations from a BLAST search?
You can. It's not part of this mapping workflow that I showed, but for example, back here in SeqBuilder, where I opened the resulting GenBank file with all the mapped features, you could certainly BLAST from here and then actually pull those annotations in. So if you wanted to do a BLAST search of a region where no annotations mapped to try to fill in the blanks, you can do that right here in SeqBuilder, which is part of the LaserGene Molecular Biology Suite.